Good evening. Good evening. Good evening. Can I get a good evening back? Good evening. Thank you. Welcome to the Shoals Marine Lab's weekly marine seminar series, our Rock Talks. I'm Sarah Morris, the John M. Kingsbury Executive Director of the Shoals Marine Lab, and I'm excited to be here on our second Rock Talk this year of the 2024 season. For those of you that are online who may not know our lab community, the Shoals Marine Lab is the oldest and largest marine lab dedicated to undergraduate education and training. And we use that term in the broadest of senses. The lab is jointly operated by Cornell University and the University of New Hampshire. Our seminars are affectionately referred to as Rock Talks because back in the day, we actually didn't have buildings and the lectures were literally given on the rocks. Today, we've moved indoors, but we still call them Rock Talks as a throwback to the history of the lab and the island. In addition to our guests joining us remotely this evening, we have 4 classes on Appledore: field ornithology, marine mammal biology, sustainable fisheries, and marine parasites and diseases. In addition to the classes, we've also welcomed our undergraduate research interns, our surges, part of the summer undergraduate research group. And some of their mentors are on island right now as well. We're excited to have everyone, both virtually and in person, here for this Rock Talk. The format for tonight's seminar will be a 45-minute talk, followed by about 15 minutes of questions and answers. For our friends online, please use the Q&A box of your Zoom screen, and we will read your online questions out loud for the entire audience. For those of you in the Commons, we'll pass around a microphone for you to ask questions to the island community. At this time, I ask Dr. Shaylee Shaw to introduce this evening's speaker.
Oh, thank you for clapping for me. I'm very excited to be introducing today's speaker, Dr. David Bonter, who is the Arthur A. Allen Director of Citizen Science and co-director for the Center for Engagement in Science and Nature at the Cornell Lab of Ornithology. He's also a senior extension associate and a senior lecturer at Cornell University. Dr. Bonter received his PhD from the University of Vermont in 2003. He has since received more than $5.5 million in funding from NSF and NASA for work involving citizen science, STEM education, and research. He is highly respected in a wide range of ornithological topics and has been named a Fellow of the American Ornithological Society, a Faculty Fellow at the Cornell Atkinson Center for Sustainability, and an associate editor of the Journal of Field Ornithology. In addition to his research and administrative roles, Dave has throughout his career demonstrated a deep passion and commitment to undergraduate teaching and mentoring. Okay, 40% of his published papers have 1 or more undergraduate authors. He has mentored 26 senior thesis students and has developed and taught more than 10 different undergraduate classes, including field ornithology out here at Shoals for 10 years. For this work, he has received a Cornell Faculty Excellence in Undergraduate Research Mentoring Award. However, what you won't find on a CV is a description of the boundless kindness and generosity with which he teaches and mentors his students. I count myself extremely lucky to have been one of those students. I met Dave over 12 years ago when I was an undergrad and interested in getting more involved in field research. Before I knew it, I was taking field ornithology out at Shoals and then spent the summer studying gulls. This experience launched a research career that I have not only Dave's scientific mentorship to thank for, but also the packs of candy he mailed in with lab supplies, the enthusiasm and encouragement that accompanied his regular check-ins, and the patience with which he helped me write and edit my first scientific paper. In a full circle moment, this is my first year back out at Shoals since I was a student here, and this time I'm teaching field ornithology. So it is my great honor to welcome Dr. David Bonter up to the lectern for a rock talk today, where he will be talking about investigating angry ants and grumpy gulls, a decade of inspiring undergraduate research at the Isles of Shoals. Welcome.
Yeah. Thank you, Shaylee. That was horrifying. What Shaylee didn't tell you when I used to send out candy, I'm getting like echoes here. Did you get an echo? Is the other mic still on? The handheld? Oh. Okay, I need to mute this. Okay, how about now? No. So what's going on? Okay. I'm gonna stay away from that. I'm still getting an echo. Well, what Shaylee didn't tell you is that when I used to send out packages of candy to the students, one year they needed a lot of hypodermic needles to take more blood samples from the gulls. And I packaged the candy with the hypodermic needles. And I didn't even think about it. That was like not like a cool thing to do. And they opened like, you sent us candy and needles. So it's like, anyway. Yeah, so great to be here. I'm still getting this. How are we gonna? Oh, I think probably what's happening is the speaker. Nope. Do that. Oh, that. All the way down. Hmm. Fine. Okay, I just oh, I think I just did. I put it onto that. Okay, all right, that's a good start. What was I saying? Anyway, it's great to be out here. A lot of my students from Cornell are here. Liz graduated 2 years ago now, and Shaylee and Grace and Braden over there. Where did Braden go? Yeah, who was going to graduate and decided not to stick around and take Shoals courses all summer. Really cool. Great to see the banders from the Appledore Island banding station. A lot of them were out here today. Andy and I go way back. Really excellent to be here. I haven't stepped on the island since 2017. It's really cool to be back. And I guess I just wanted to start off here. If I can move the advanced slides now. There we go for this photo. Let's get rid of this. Get rid of this. Good. Zoom. Is that better? Okay. All right. So the reason I came out here for the first time was just this really random event that happened in Veracruz, Mexico. I was at a conference, a big bird nerd conference in Veracruz, Mexico, at the opening reception, and I just happened to run into my old friend, Sarah Morris. And at the same time, my boss at the time, the director of the Cornell Lab of Ornithology, John Fitzpatrick, was walking by, and Sarah grabbed Fitz and said, I'm not gonna be teaching field ornithology at Shoals Marine Lab anymore. Dave should teach it. And it was just one of those like random things that happens in life that completely changed the next decade of my trajectory. I came out here, started teaching, started working with undergraduate students more, started mentoring amazing students, and it was all because of that random cocktail party run in Veracruz, Mexico. So if those sorts of things happen, run with them and good things can happen. Not advanced. There we go. Another class back here. Some of you might remember or might recognize the person in the circled in the yellow there. My first year on the island, Dr. Kristen Covino, faculty for field ornithology, was actually working down at the banding station. So we all keep coming back. But tonight I'm just going to talk about, give you a whole series of stories about students that I've worked with over the years, and all the work that I'm going to show today, it's the students. So pretty much everything I'm going to show is because of the students, and also I advise a lot of students in the environment and sustainability major at Cornell, and a lot of the students are constantly wondering what's next. Like, what could I possibly do with this major? What am I going to be doing 5 years from now? So for each of the stories I tell you about these amazing students that I had the opportunity to work with out here, I'm also going to tell you where they are now. So maybe you can start thinking about some of the possibilities down the road. And all of the students that I work with, I really like them to come up with research questions, and those research questions often start with an observation, right? They're out here at Shoals, usually taking a course like field ornithology. They see something, they start to wonder about it, they start to ask questions, and then they return to do a summer-long research project and answer that question. So starting off with Michelle Moglia, the one in front here, the one in back you also might recognize. And Michelle, when she was out here in the course, she noticed walking around the island and looking at all the gull nests that there's a high degree of variability in the coloration of the eggshells, right? So great black-backed gull nest, the typical eggs there, like the one at the top, that really deep green background color with a high degree of speckling or maculation on it. But even within a nest, and all of the eggs within a nest, in theory, should be laid by the same female. So that same female is laying these eggs that have very different coloration. Why is that? Sometimes they don't have any of the coloration at all, or very pale eggs, and other times weird things show up in nests. So Michelle was really interested in this, and all of the coloration that happens in eggshells globally is due to two different pigments and different proportions of those pigments in the egg. The protoporphyrins, which give the dark colors, the browns and the blacks and the speckles, and biliverdin, which gives the greens and the blues. So it's just different combinations of those that determine what an egg looks like. And here's another series of great black-backed gull eggs and the variation that you can find in nests here around Appledore. We don't know really why eggs come in these different colors and patterns and shapes. There are a few hypotheses. There's the camouflage hypothesis that suggests that this maculation and these dark colors help the egg blend into the background and make it less likely that predators will eat them. There's a sexual signaling hypothesis. These pigments are very expensive for the birds to produce, for the females to produce. And in theory, a high quality female would be producing more of the pigments. And in a system like the gulls here, both the males and the females have to be contributing in order to successfully raise the young. So if the male's looking at the eggs and being like, wow, the female I mated with is really high quality, she's putting a lot of pigment in those eggs, he might invest more. But first we need to come up with some way to quantify the coloration of eggs. We can look at them and say, okay, that's pale, that's dark, but how do you actually quantify it? And poor Michelle spent a couple of weeks in this pop-up tent out in the colony with me and a computer and a spectrophotometer to measure the color of the eggs. And we had a ton of fun in there. Shaylee was getting pooped on, grabbing all the eggs and passing them through this window to us, but it was a lot of fun. And what a spectrophotometer does is show you the amount of light that's being reflected there on the vertical axis at different wavelengths along the horizontal axis. Okay. And in our visual system, we see all the colors of the rainbow, right? So from purple all the way up to red, everything in between. Birds are super cool in that they actually have another cone in their vision, and they can see way down into the ultraviolet portion of the spectrum. So birds are seeing things in the world that our brains can't even begin to perceive. If you take a look at this graph here, these are 3 eggs, this spectrophotometry measurements of 3 eggs from the same great black-backed gull nest. You can see that egg 1 and egg 2 are basically the same. Those lines are sitting on top of each other. Those eggs look alike. Egg 3 is reflecting a whole lot more light back to the device, which means that that's a very pale egg. And what Michelle found in studying all of the eggs in 84 different nests out here on Appledore Island is that third laid egg was often much brighter, much less pigment than the first and the second laid egg. That's what that graph there shows. So who cares? Third egg is lighter. Does it have any effect on the reproductive success, on the fitness of the nestling hatching from that pale egg, right? In order to get at this, Michelle went to every single one of those nests every single day during the season in order to find out which chick hatched from which egg, marking them as they're hatching on the tip of their bill. So she knew the hatch order and which bird was tied to which egg, went back, measured those birds on a daily basis to get their growth rates throughout the nesting season. A whole lot of work, ton of work. And she found out a whole lot of nothing, right? So basically all of the traditional measures of reproductive success, egg volume, hatching probability, the weight of the nestling when it leaves the egg, and the nestling growth rates did not vary depending upon, it was not related to the color of the egg. So that doesn't necessarily mean, well, evolution works in strange ways, right? The pigment could mean something somewhere else. So there could be spatial variation. It could have meant something thousands of years ago, but it's just a relic now. But basically what Michelle was determining is that these birds were running out of ink. Those pigments, they were putting them all into their first and second eggs. And by the time the third egg was laid, they were done and the eggs were much brighter. So this is Michelle today. After she left us here at Shoals, graduated from Cornell, she went on and got a MBA in conservation finance at UNC Chapel Hill. She's an investment banker in New York, and she just invested a whole lot of energy in that little one there named Avery, a little cutie. Far less adorable than Avery, Matt Savoca. All right, and Matt would be the first one to say that. So Matt was one of my first interns out here. Took the class, TA'd the class, and then came back for another summer of research. And Matt's question was really focused on the observation that some gulls nest in really weird places around here, right? You've got the dense gulls out in the colony, and then you've got birds nesting under Palmer Kinney and in drain pipes and random places around campus. So there are essentially two nesting contexts for the gulls on Appledore Island. Those that nest in dense colonies, mostly the rocky periphery of the island, where the nests are in general less than 5 meters apart. So dense colonies. The loose colonies are these birds mostly around campus, near buildings. Nests on average are greater than 20 meters apart, right? So why do the gulls do that? There are a lot of theories. The costs and benefits of colonial nesting have been debated for decades. It could be that some of the costs could be that diseases are transmitted more quickly out in these dense colonies. Think about freshmen in dorms, right? Disease transmission. But some of the benefits of colonial nesting is that you can be sharing information about good food sources. And one of the big ideas is that there's value in that group defense, right? So for those of you who've been over to White Island and seen the terns, and you walk through the tern colony and they're all attacking and driving you out, or if you've been out on the rocks here, as a potential predator, when those birds all start flying at you, you want to get out of there, right? They start screaming, they start diving at you, they start pooping. That community defense is quite effective. It's really interesting in this system, though, particularly with the herring gulls, because the great black-backed gulls that are nesting here are also, while they can help protect against mammalian predators, those gulls are also likely to eat your young, right? So it's an interesting system. So what Matt did was try to quantify nesting success in the loose colony and in the dense colony. He had hundreds of nests all around campus where he marked them, measured the eggs, checked growth rates, nestling survival, and so forth on a regular basis. He found lots of nothing to begin with. No difference in egg size, no difference in hatching probability, the number of nestlings hatched, the weight of those birds at hatching. But, this is a big but, survival to fledging was very different. So the birds out in the dense colonies were far more likely to survive and leave the island than the birds nesting around the buildings. Okay. Big result. Matt published that as a senior thesis, got into UC Davis for his PhD, where he went on to study whales and plastics in the environment. Has a whole series of really high impact papers now in Science and Nature, and National Geographic Explorer. and has quite a good career, even though he left birds behind. So Matt found out that the birds in the colonies tend to survive better. Why on earth would any birds choose to nest around the buildings, right? And this is what leads into the idea of these long-term studies can be really quite valuable. We had interns out here every summer for 10 years, marking nests, monitoring reproductive success in the same way every year. And if you were out here on June 4th, 12 years ago, you would have been in day 3 of a really, really miserable nor'easter, where it was about 52 degrees, really strong winds coming in, horizontal rain, just a miserable couple of days. It was awful trying to teach during that period. You couldn't go outside. So this big, big nor'easter storm, waves were hitting the lighthouse over on White and Seavey. When it all ended, we went out into the colonies, and all of those nests that we had just spent an entire week marking and measuring the eggs and so forth, we had lost a whole bunch of them. A lot of them had just been washed away. And where did we lose those eggs? Out in the dense colonies around the periphery of the island. Okay, so the red dots on the map were all the nests that were destroyed during that intensive storm. The blue dots all survived the storm. Okay. So in that particular season, being in the dense colonies around the perimeter of the island was actually pretty bad. You look here at the proportion of the nests that are surviving as the season goes on. The blue line there is the dense colony, really bad in 2012 to be around the periphery of the island. So in some years it could be good to be in the loose colonies. Some years it can be bad, right? That's why that persists. Here's another way to look at it. Nest losses by year from 29 to 2012. That yellow line was the year with the storm. So you can see that huge impact that one storm had on gull reproductive success in that year. And because we had this long-term data set, we had students collecting data in the same way every year. Shaylee and Michelle, who you just met in the last story, and Sarah McLean there on the right, were able to write this paper about how the optimal nesting sites can change over time. All right, shifting now to some animal behavior research featuring Shaylee. But first, Sarah McLean there on the left. And Sarah started off with the observation that these gulls react very differently to various kinds of threats. And you've probably noticed this yourself walking around campus. You might be walking by a gull. If you're pretty far away, it might just sit there. It might go, like it doesn't really care. And you walk on by, no problem. But if you walk a little bit closer to it, it might stand up. It might give you the eye. It might start doing this kek, 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 kek warning call. And then once it takes flight, that's when you know, okay, you need to get out of there, right? So there are various stages in which birds respond to these threats. And Sarah was really interested in well, we know walking towards a bird, it's a clear intimidating, I'm intimidating you, Grace, right? React, right? But what other cues do they key in on? Can they hear things and react to it in an appropriate way? So Sarah set up this really cool but simple sound playback experiment where she put speakers near nests, went far away, and played a whole series of different stimuli to the birds and quantified how they reacted. One of those stimuli was the song of a song sparrow, very common little songbird on the island, completely non-threatening to gulls, familiar sound, right? Another one was western scrub jay. This is another smallish bird, would be zero threat to the gulls on Appledore Island, but it's a species found way out west, and the birds here likely have never heard that vocalization, right? So it's a novel sound to see how they would react. She played back alarm calls of herring gulls and great blackback gulls to see how they would respond. She played back a recording of the human voice, and this was Sarah reading Jonathan Livingston Seagull. She thought that was cute. So do they respond to the voice of a human? And then the other is a potential predator, the vocalizations of bald eagles. And after doing a whole series of playbacks where she scored the responses from 0 being the bird sat there and did nothing, to 8 being the bird went ballistic and went into full attack mode, she found out that birds really didn't care about the song sparrow. right? That's what you'd expect. That's kind of our control in this experiment. They were mildly curious about that Western scrub jay, that WESJ there, that's Western scrub jay. So they're mildly curious about that. It was a different sound. They didn't know what it was. They paid attention. They looked around. They might even have stood up. They responded quite significantly to alarm calls of herring gulls and great blackback gulls. A little less to the bald eagle vocalization, and that kind of makes sense because when a bald eagle is attacking, it's usually not vocalizing, despite what you see in the movies. You know, they don't announce that they're attacking. And the human voice set the birds off, really irritated them. So the birds basically were responding to auditory threats exactly as you would expect. So they're keying into these threats as you would expect. Sarah published that. She actually published 3 papers from her undergrad work out here. And she is now a professor. She ended up getting her PhD in conservation biology at Berkeley. She's now a professor out in California, but that's kind of her second gig because the whole time she was out here and even before she came out here, she was writing fantasy novels and she signed a 6-book deal with Penguin Press. And her first book is coming out like next month, right, Shaylee? So read The Phoenix Keeper if you're into sci-fi fantasy, and there are 6 more books coming in this series. All right, next up, so Shaylee was out here as a student and working with Sarah and was, I think I can say, inspired a bit by Sarah's work. And what Shaylee realized is that that yow call that you probably have all heard from the gulls can sound different at different times, right? And birds use vocalizations to communicate a wide variety of things, right? So you hear a bird song, and often it's to attract a mate. It's to say, hey, this is my territory, you stay out. They use contact calls to announce their presence to other birds, begging calls to say, hey, I'm hungry, bring me food, and then alarm calls to communicate a sense of danger. And what most people don't realize is that birds can actually alter their alarm calls to encode information about the kind of threat. So a chickadee gives a certain alarm call if it's a snake and a different alarm call if it's a hawk, right? But what do the gulls do? And here are 2 spectrograms, they're basically just ways to visualize sound recordings. The one on the left there is the call, the alarm call that a herring gull gives under a low threat situation. Okay, sorry about that if that's a little loud. All right. And visually you can see, so that graph shows the visual representation of that sound, right? Frequency over time. The one on the right looks different, right? Let's see if you can hear the difference in the sound. If I can find my cursor. Where did my cursor go? Okay, there's a little bit of a break in it. That's the low and the high. Okay, so you might be able to hear a little bit of difference there. Okay. So Shaylee was wondering if that was actually encoding what we thought it was encoding. Was it encoding the degree of threat? And I really should have Shaylee doing this because I'm probably really messing it up. But she set up a interesting playback experiment. Again, a speaker near a gull nest, video camera, played back a whole series of different stimuli, that high threat call at a high frequency. So yow, 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 yow. The bird's really irritated. High threat at a lower frequency, low threat at high frequency, low threat at a longer frequency, and then the song sparrow control again. And the results here were also, again, really clear. Song sparrow control, birds, yeah, okay, no problem. That low threat level call, the focal birds noticed, they reacted a bit, but it was that high threat call at a high frequency level that really set them off, right? So these birds are clearly encoding information about the threat in their alarm calls. Shaylee published this in Animal Behavior. One of a couple of papers from undergrad, went on to do a PhD at Columbia, studying superb starlings in Kenya. Really cool place. I got to take students there to work with Shaylee, and that was one of the highlights of my career. Did a postdoc at the University of Rochester, is now a National Science Foundation postdoc at the Cornell Lab and faculty at Shoals Marine Lab. And one of the other highlights of my career is I got to be the only witness at Shaylee's wedding during the start of COVID. So I was the only guest and the witness at her wedding. So it was fun. All right. So this is Taylor Heaton. Taylor was out here started out here, took the class the year that you were doing your research, and then came back the next year because she made the observation that she thought that the parents at gull nests changed their behavior throughout the nesting cycle. And she wanted to quantify it and understand what was going on there. So all of your parents would probably say, we invest an awful lot of energy in our offspring. We have a limited amount of energy to invest. And in the bird world, defending offspring is a risky endeavor, right? You might save your offspring, but you're also putting yourself in a position of risk, right? So they want to be smart about it. They don't want to just defend, right? You can't defend with unlimited resources. There are a couple of ideas here about when adults should invest in defensive behaviors more. One is this offspring vulnerability hypothesis, which suggests that parents will invest in nest defense most when the offspring are most vulnerable, right? Little guys need help, parents react. The offspring age hypothesis suggests that parents should invest more in their offspring's defense when they're older, right? I've already invested a ton of energy getting this kid to be a teenager. The last thing I want to have happen now is for them to die, right? So invest more later on, okay? And we do know that the riskiest time for these nestlings is in the first 10 days after hatching, right? So this is a survival analysis that shows the probability of survival on the y-axis at different days of the life of the nestling, right? So that day 1 to 10 is the red zone. That's where a lot of these offspring die. If they make it past day 11, then they've got a pretty good opportunity to make it to fletching, right? So what Taylor did was conduct a whole series of simulated threat experiments on hatching day and at different stages during the nestling's life and quantified how the parents reacted to that, okay? When the birds are incubating the clutch, they were pretty chill. They didn't really react heavily. The dark bars there, the high level of reaction, the gray medium reaction. So they were pretty relaxed at the clutch stage. The day the nestlings hatched, the parents freaked out. Right? You go up to the nest and they get really, really aggressive. Dave Buck's laughing back there. He probably has personal experience with this, right? Yeah. But then as the chicks grow a bit older, start becoming mobile, start moving around a bit, the parents relax, okay? So this supports that offspring vulnerability hypothesis that the parents should engage in more aggressive defensive behaviors when the nestlings are young. Taylor published that in Ethology, is currently a zookeeper at Oakland Zoo out in California. And I just talked to her last week and she's starting at vet school at Colorado State in the fall. So really excited about that. Okay, Luke to Fisher. Luke's observation literally bit him in the ass, okay? If you sit in the vegetation around the island here, you might become familiar with these guys. Myrmica rubra, the European fire ant. This is an invasive species of ant that at some point in the last 100 years or so was introduced here on the island and at various places along the seashore here. They form very big colonies and they're quite aggressive ants, right? So Luke sat down one day, got bitten, sat down on one of these ant hills here on the island, just right up there, got a lot of bites, and then started to pay attention to what the ants were doing. He started to notice some gull chicks that were swarmed with ants and dead, right? And so he was wondering what effect these ants have on the reproductive success in the gulls. So Luke went out, marked a whole bunch of nests. Every day he went to the nest to figure out if there were ants in them or if they were ant-free, and related whether or not the nest had ants to the growth rate of the chicks. So the nestlings in nests that did not have ants grew at a significantly faster rate than those in the nests with ants. He found a number of cases where as the nestling is just peeping out of the egg, ants start to invade and kill the nestling before it can actually hatch. And other cases where, they hatch out, they're kind of tired, they're not moving around much, and the ants swarm them and kill them. Fire ants are known to attack nestlings in a whole suite of different birds, but Luke's study was the first to show this in gulls out here on Appledore. So he published that paper, and Luke ended up becoming a manager of a cider distillery at his family farm in upstate New York. Okay, are we tired of gulls? I'm tired of gulls. We're going to switch to warblers now. Okay, and this is a story about migratory connectivity, and that little bird there, yellow warbler. They're all over the island. If you haven't seen them yet, find one of the field ornithology students and have them show you this bird. It's really cool, pretty little bird. You should know it. Migratory connectivity is really about understanding connections between breeding areas and wintering areas in birds, right? If a population of birds is declining, it's very helpful to know where they're spending the winter, right? And for a lot of these small birds, it's pretty tricky to understand, to draw those connections between breeding areas and wintering areas. They're really tiny. You can't put big transmitters on them. It's a hard thing to study. So I'm gonna let Max tell you what he did.
All right, I'm Max Wojcinski. I've been attaching geolocators to yellow warblers here on Appledore Island. They're like this. The light stock records ambient light levels over the course of the year, and if we recover this bird next year, we can use that data to figure out where he spent the winter. So it's pretty exciting.
Right, so that's Max. And another thing you need to know is that these birds are highly faithful to their breeding territories. So when we catch a yellow warbler by the shoe tree here one year, if that bird survives and comes back, it's going to be nesting by the shoe tree again next year, right? So they're incredibly faithful, which kind of blows your mind, right? They fly all the way down to South America and they come back and nest in the same bush or near the same bush year after year if they survive. So that's the geolocator there on the back of the bird. We have to recover these. They're not transmitting. They're super tiny, super lightweight because a yellow warbler only weighs what, 12 grams, Dr. Moore? 10, 11, 12, they're fat. Yeah. So Max put out a bunch of these transmitters on birds here on Appledore, and then also near his home in Minnesota, Wisconsin area. And we let the birds go and hoped that we got some of these transmitters back, which we did. And here are the tracks. So the Appledore Island nesting yellow warblers are wintering in Western Colombia, mostly west of the Andes, whereas those birds in southern Minnesota are wintering in Venezuela. So we have this really strange crisscross pattern where the westernmost breeding yellow warblers are breeding east or wintering east of the eastern breeding birds. So that was pretty neat. Max's paper was actually the best paper in the Journal of Field Ornithology that year. Won an award for that. Max went on to get an MA in science communication at the University of Wisconsin. He's currently a PhD student at the University of Michigan, where his advisor is another one of my former undergrads. So I'm feeling really old. All right. And one more smaller bird, quick study here. This is Facundo Fernandez Duque. If you've been down in the grass lab, the shop behind the grass lab there, you might see some barn swallows nesting, these little mud nests up in the rafters. There are also a bunch under Palmer Kinney. Any others out here anymore? Or there used to be. I don't know. Are they still under there? Okay. Yeah. So these barn swallows build these mud nests, and they take a lot of energy to build. So they reuse the same nest year after year if it's still there. And that means that they can build up loads of parasites. And next to the dime here, you see all those little specks. Those are mites that live on birds. And these nests can become incredibly full of these mites. And Facundo was, banding barn swallow nestling, and there are just mites running down his hand as he's banding these birds. And he wondered, geez, does this have any sort of effect on the birds? And yes, it actually does. So nestlings being raised in nests with a high mite load are growing at a much slower rate than nestlings in nests that have very few mites. And in the world of small birds, we know that the weight of that nestling when it fledges is highly correlated with survival probability, right? So fat nestlings, when they leave the nest, have a good chance of living and returning. Thin nestlings don't have as good of a chance. That photo there are two nestlings. The lower one is from a nest without mites. The upper one is from a nest with mites. Those birds are the same age. You can see what a big impact that can have. Right. Kundo's now, so Kundo was my last intern out here in 2016. He's now working on his PhD at the University of Illinois, doing a cool project there with red-winged blackbirds. And so I guess overall, in a decade worth of working with students out here, we had a lot of fun. We got pooped on a lot. And we ended up getting a number of scientific publications, which ended up being the ticket for a lot of those students to get into grad school when they have a publication out of undergrad. A couple other students I just want to mention. Not all the students publish papers. We worked on different projects over the years, but they're doing really cool things. This is Christine Wilkinson. Christine's a superstar. She's going to be president someday. National Geographic Explorer, got her PhD at Berkeley, studying hyenas. She just had an episode on Peacock TV. Christine does a lot of research about homosexuality in the animal kingdom. So there's a show on Peacock about this, featuring Christine. And she also started the Black Mammalogists movement, which is a pretty intensive thing for her to be working on. Justin Stillwell was one of my first interns out here. We lost him to the dark side. He's an aquatic pathologist now, but he ended up getting his PhD or his veterinary degree at Georgia, professor at Mississippi State. Nobody could take a blood sample from a gull like Justin. Liam had a really cool project working on eiders out here. He's wearing that suit. He spent so much time walking through the rose bushes and the poison ivy. The kid was immune to it. Between K House and Smith's Cove, he found 111 eider nests one year. So there's a big concentration of eiders nesting. When you get off the dock, off to the right there, those bushes are just full of eiders. And right now they're all hatching. So those of you who just got on the island, you'll start seeing a lot of these fuzzy little eider ducklings hitting the water, and hopefully not too many gulls eating them. Another student who studied eiders out here, Ben Shattuck, spent a lot of time playing his banjo under the shoe tree. Ben's carrying on the artistic and the writing tradition from Shoals Marine Lab. He went on and got an MFA in writing. Has published his second book just came out. It's on the New York Times bestseller list. And he also married Jenny Slate, the Saturday Night Live actress and comedian, and they just had a kid, so he's doing well. Yeah, so I guess in sum, I just want to say, like, take advantage of those random opportunities in life, right? If I didn't run into Sarah at that conference, I probably never would have stepped foot on this island, never would have worked with amazing students like Shaylee. And yeah, be observant, right? As you're taking these courses, walk around, ask questions, notice stuff, and then come back and study it. It's a cool opportunity, right? All right, just want to thank a bunch of folks. The staff at Shoals every year is phenomenal. I don't know the new staff very well, but I am assuming you are equally as phenomenal as it's always been out here. So much that happens out here can only happen because an incredible staff. So be sure to thank the captains and the engineers and the folks who help keep this island floating. Really enjoyed spending a lot of time with the Appledore Island banding station folks over the years. So Andy, please please tell the whole crew how much I appreciate hanging out with them. A whole bunch of folks for funding and such. And I don't know, I've been rambling. If there's time for questions, I'd be happy to take it. All right, right on time. How are you doing?
All right, we do have 15 minutes for questions, so I will bring the microphone around. If you've got a question, please raise your hand. I don't believe we have any online right now. We're looking.
All right, thank you very much. It was a great talk. I was just wondering, did you ever figure out why those two populations of birds were crisscrossing their migration?
No, and everybody was really curious about that. Yeah, there are a few other species where you have this sort of crisscross migration, but also we could see the paths from those devices too. And a couple of birds went down through Florida. A couple of birds did this trans-Gulf migration. So they leave the coast of Texas and fly nonstop until they hit the Yucatan, which is pretty cool. So yeah, they're all doing different things and going to different places. So yeah, who knows why, but
So earlier you had, thank you for your talk, it's really interesting. Earlier you had said that if you were disrupting the gulls, you may get a lot of poop on you, but I thought, and I'm probably wrong about this, that birds don't really have a way of telling themselves when to use the bathroom. But is this like a defense mechanism or is it also random?
it I don't know whether it's random or not, but it is a very effective defense mechanism. And I challenge you to walk through the colony 2 days in a row and not get pooped on. Yeah, it's yeah, and like and eiders too. So eiders nest on this island, and when they're scared off their nest, they leave behind the secretion that is the most putrid smelling stuff over the eggs, and the idea is that might deter like a fox or a predator that might want to eat the eggs. So they're doing that on purpose when they're flushed from the nest.
And there's a long history of the way we've talked about it is physical and chemical warfare. And there's no question that they get much better with their aim during the course of the season.
Yes. Yeah. If you go out in the colony, go out in the morning before they've all eaten, and they have less to share. If you go out around dusk, you're gonna come back looking pretty bad and smelling pretty bad. Fair one. Good. I talked through the sunset. Sorry, folks. Got a question back there.
I know you kind of like already talked about, also thank you for your rock talk. But as somebody who wants to also, or as somebody who wants to work with birds in the future, what would you suggest like in the next steps, like trying to go to grad school and stuff like that for it?
Yeah, that's a big question. So I think it's the ornithological research community is pretty small. So making connections is really key. If you have an opportunity to go to a conference, Wilson Ornithological Society, Association of Field Ornithologists, American Ornithological Society, and absorb talks and meet people and make connections, that's a big one. Another thing that a lot of folks do is take a year or two after undergrad and do field jobs. Go and help with the conservation of some honey creeper in Hawaii for 3 months, and then go down to Costa Rica and do something with mannequins, and then go to Florida and do something with waders. And you'll make a lot of connections in doing that. You'll gain some really critical field skills that potential advisors would want you to have, that would make you more attractive to potential advisors. And I think it's also important to realize what you don't want to do. A lot of that stuff sounds cool. And then, you go and spend a summer up in the Arctic, working with shorebirds, and realize I hate mosquitoes and I never want to be working in that system. So yeah, I'd say take a year or two off. I think too many folks are going straight from undergrad into PhDs now. and they're, 28 years old with a PhD and they've only have experience in one system. I'd really like to be working with folks who have a lot of different experiences. So, Dr. Morris, you're the best mentor I have ever known. How would you answer that question?
You did a great job.
She punted. Yep, got one here. For Zoom.
So I was really interested in the yellow warbler study.
It was the
Um,
coolest.
Yeah.
Sorry, Kaylee.
And I was just kind of wondering, like, how are they able, or like, do you know how they're able to like travel all the way back to where they originally came from? Because like I know personally, I need like Google Maps for everywhere. So I'm just like wondering how these like really small birds can just like fly across continents and like get back to where they came from. 'Cause it's really cool.
Yeah, how birds navigate and orient has been the topic of research for hundreds of years. And basically the answer is there are a whole lot of different ways. There are magnetic cues. So we live within this, like the Earth has a magnetic sphere and some birds can sense that. And so they know where they are on the globe based on magnetic cues. There are some olfactory cues. There are some visual cues looking at the patterns of the stars. And all of this in birds with a brain the size of a pea, which it's really remarkable. And they can fly hundreds of miles in a night. So the birds stopping here on Appledore are engaging in something called hyperphagia. They're eating like crazy. They're putting on layers of fat, waiting for good wind conditions, taking off, flying another couple hundred miles towards their destination, stopping, burning through all that fat, stopping there, feeding again, doing that next leap. So it can take them 6 weeks, 8 weeks to go from Venezuela to Appledore Island. And how they exactly get to, the shoe tree to build a nest. Look at Sarah studies migration too. So, yeah, there are a lot of theories. There's a lot of evidence for a lot of them. There are a lot of people in the field who get really worked up thinking that their theory is the best. I think that birds are using multiple cues and multiple systems. And it's a massive selection mechanism. So on the coasts, East Coast, West Coast, during fall migration, almost all of the birds are juveniles. Many of those birds are probably don't have finely tuned orientation mechanisms, and they're ending up out in the ocean, which means they're not contributing to the gene pool.
So you were talking a lot about like making connections and where people are now. And I wanted to know what's the best advice you've ever gotten, whether that's professionally or not.
Oh boy. Best advice I've ever gotten is you should teach your field ornithology at Shoals Marine Lab. Now, another thing I didn't mention is reading. The scientific literature is really important for a few reasons. First off, and actually don't read, skim, right? So find a whole bunch of different scientific journals, conservation biology, biological invasions, ornithology, mammalogy, whatever. Skim through titles, skim through abstracts. If you're bored, keep going, right? If a title catches your eye, read the abstract. If you're still interested, keep reading. That's how you really realize what gets you excited. You want to do something that interests you. You don't want to be stuck studying something for the next 30 years that you are kind of lukewarm about, right? So that's the first reason I think to read, is to just figure out what your area is. And if you start that skimming process, it should become clear pretty quickly. Once you know what you're interested in, all of these fields are relatively small. You'll start seeing the same names coming up over and over again, right? If you're interested in migration ecology, you're going to see Sarah's name on a whole lot of papers. You're like, maybe I should work with Sarah, right? Send an email. Don't just say, hey, I like birds. are you taking grad students, elite, right? Be like, hey, I just read your paper on common yellowthroat, blah, 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 blah. Found this was really interesting. Have you considered X? You know, and that gets you in the door. So, yeah, I'm not saying pick up a scientific journal, read it cover to cover. Nobody ever does that. Don't get bogged down in the stats, any of that stuff. It's about the questions, about the study systems. Yeah. Does that help? Okay. Grace, I'm scared. I'm really scared. Grace has been in my lab group for a couple of years and Grace could be ugly.
Anyway, thank you for your talk. But I had a question about the sub-colonies. So is there a high preference for being like on the perimeter as opposed to inside? Because like if you think about like the ideal, would it be more inland and in a dense colony?
Oh, that is in Matt's paper and I can't remember exactly what happened. I know in some colonial nesting species, being at the core of the colony is really important. And being around the periphery, you're more exposed to predators. It's probably less optimal habitat. I can't remember what Matt found here. One of the problems here too is that the colony is kind of linear, right? It's not like we've got a nice perfect circle of an island like over on White and Seavey where it is more of a circle and you could be at the core of the colony. Here, you know, it's the rocks are linear, so there's no like really central area.
Or I guess more just asking like there's something about like being on rockier edges that's like very preferable for them aside instead of being like in the grassy areas where it is more inland, but you would be, I guess, more protected from like inclement weather.
Yeah, I think it really depends season to season. Like it's a hot spring, right? And the birds out on the rocks today on June 4th were panting like crazy. They're overheating, right? During a normal spring, they're not stressed because of that. So this might be a year where having a nest in the shade could actually be better. And then you throw in crazy things like invasive ants that are only a problem around the buildings, right? Out on the rocks, the ants aren't there. There's no soil for them to live in. So that's something that's changed in the last couple of decades, right? You know, 100 years ago, gulls on this island wouldn't have had to worry about fire ants. So nothing stays the same. Things change from year to year. You know, we might think we have the answer, but then conditions change.
Thanks.
We have time for one more question.
Bedtime.
Yep.
Thank you for your time. I was just wondering about your thoughts on whether you think there are limitations to understanding gull behaviors because we can't fully comprehend the UV scale that they can. And if you think at some point in the future that we'd be able to connect like patterns of their behavior with their sight, with what they can see and we can't.
Yeah, there are so many questions around the visual system and what's going on in the ultraviolet spectrum. There's an amazing collaborator. I think I had her name on the thank you slide there, Kazi Stoddard, who's, yeah, if you're looking for a grad advisor, Kazi Stoddard would be phenomenal at Princeton. Does amazing things with bird vision and trying to come up with ways to understand how birds see the world. And there's you know, like you think about a black-capped chickadee. If folks know a black-capped chickadee, it's a little bird. They all look the same. Males and females look alike. They have this silly little black cap. In the bird vision system, on the males, that black cap is glowing, like in the UV portion of the spectrum. So to our eyes, males and females look alike, but to chickadees, they're incredibly different, right? So they're seeing this world that we can't even begin to perceive. I think there are a ton of questions in that area. Yeah. Cool.
So I want to thank everybody for tuning in this evening, those that are on Zoom, as well as those that are here in the Keegan's Comments. I want to thank David
Oh, was I
Bonter.
supposed to be standing in front of the camera?
A little bit.
Oh, sorry, folks.
I want to thank David Bonner for coming back to Appledore after a too long absence. And I think one of the things that you're seeing is the long history that Shoals has in providing educational opportunities, both in terms of classes, but also in terms of the mentoring and the research opportunities. And Dave has done a phenomenal job in highlighting what students have done and then where they have gone using some of what they've done. So thank you again so much, and tune in next week for Rock Talk number 3.